Hi guys. Today I'm going to talk about something that we're developing in our lab. And we, like Dr. Gallagher and Dr. Shu said, believe that there has to be a lot of shots on goal, basically because we're getting into dire straits with all of the clinical trials that have been failing. And B, there's an untapped repository of about 12 to 15,000 FDA-approved drugs. And we think that there's no such thing as side effects, there are just effects. And so I'm going to discuss a pipeline, computational pipeline in our lab that we've developed to screen these drugs for use against Alzheimer's disease. So we've decided that in the age of big data, we can allow the brain to tell us what it needs as opposed to trying to figure out what it needs from the literature. So the way that we do this is we apply integration techniques to large data sets, which come from the GEO portal as well as the AMP-AD portal. And we're looking for data sets in our lab coming from the temporal lobe because we're most interested in the epicenter of AD. And we're also interested in data sets that have ApoE-4 genotyping, as we believe strongly that ApoE-4 is a large risk factor for the disease. So we take these data sets and we stratify them by ApoE genotype to get a consensus disease signature across data sets for what genes are up or down regulated in disease. So as you can see, I have the sample sizes here that we're working with currently. We have a pretty robust sample size after meta-analysis for E33 and E34 disease. But the bottleneck comes in E44 controls. These people are very hard to find, mostly because most E44 people get the disease. But we found across a few data sets, we found an N of 7, which is the largest N we've seen so far. And then we do a special rank-based meta-analysis approach to get a genotype-specific gene expression signature. And what is most striking to us is even with this really, really small N, so quite small power, we're seeing these large differences in these E44 individuals with AD, suggesting that E44AD is actually quite different on the transcriptomic level. So then we are leveraging computational drug repurposing tools that are publicly available. So we're working with a tool called the Connectivity Map, which was created at the Broad Institute. And this is a compendium of 13,000 compounds that are analyzed via transcriptomics. So they're looking at their network effect. We're not looking at one pathway, we're looking at a complete network effect of a molecule in cancer cells for this data set. And this network effect will give us all of the genes that are upregulated and all of the genes that are downregulated by these 1,300 compounds. And our hypothesis for drug repositioning is that a compound that changes the disease signature back towards a healthy state is hypothesized to be efficacious in disease. So we take our compound which most flips the disease signature back towards healthy normals, and we have that as our compound A, or top compound. So we see here I have a diagram of what this compound A does at the gene expression level, where you can see the downregulated genes in red are a lot of them are upregulated and vice versa. Obviously, it's not a perfect correlation, but nothing in bio is perfect, as I've learned. So here are the scores. So the connectivity map scores. Negative score is highly anti-correlated or highly flipping the disease signature. And a positive score means you're mimicking the disease. So we're looking for most negatively scored compounds. Our first compound here has a very low score in E44AD, and it's also negatively correlated in E34 and E33 disease, suggesting that it might have efficacy in these diseases as well. So now in our lab, we're working on validation. So we've popped it into a couple of mouse models, including our lab is most interested in ApoE-4 mouse models, which we found efficacy, which is very exciting. And we're looking at RNA-seq strategies to validate the mechanism of action of this drug. Because we've come from such an unbiased approach, we now actually don't, we have ideas about why the drug might work, but we don't have a completely validated mechanism. And then we're looking at kind of computational ways to figure out how the drug crosses the blood-brain barrier, how to select correct dosages of this drug as well as other top candidates, and then what behavior studies we should be doing. So yes, in the future, we're looking to continue our genotype-specific computational approach, leveraging big public data. We're hoping for more data sets to come out with more of these E44 controls, because those are really our key material here. And then we're also exploring combination therapy prediction methods, so things that take 2 or 3 compounds together to completely combat the network signatures of disease that we're seeing. So thank you so much.
Just briefly, a quick reminder that we are video casting, and just to speak close to the microphone, because apparently there has been some interruption in the sound, but just please remember people are listening out there.
So we do have some time. We would like to thank everybody for really stimulating presentations and open up for discussion and questions.
So I'd actually like to kickstart this, and following up on an issue that Linda brought up, and thinking about what, if the panelists, any of the panelists have an idea around what biomarkers would accompany their therapeutic development for an indicator, an early indicator of efficacy or lack of efficacy, or responders versus non-responders. Michaela, do you want to?
Yeah, well, got it. Thank you. So our trial using levetiracetam has a very strong biomarker component to it. I work with people at Johns Hopkins who've got good, very strong signals for neuroimaging changes in the entorhinal cortex where the early pathology spreads. And so what we're doing is incorporating into the trial a sub-study of a smaller number of patients that will actually use tau imaging along with structural imaging to look at progression. I mean, what you really, really want to do is to find biomarkers that more efficiently, with greater power, predict clinical outcomes. This is a phase we're in, and we haven't actually settled on them, but there's a very good reason to look at the effects of a drug that decrease neural activity, which has been shown to cause the spread along the tau pathway.
Terrific. Linda, do you have a blood-based approach?
So because the neuroinflammation imaging biomarker is really not very good, sorry for those imagers out there, and it's really unclear what exactly it's measuring in terms of inflammation. We've been focusing on fluid biomarkers, and so we're planning to use a panel of inflammatory markers because we don't want to pick just one cytokine, because there are lots of different cytokines, both pro-inflammatory and anti-inflammatory, and there are now these nice inflammatory panels that you can use. So we're planning to use both CSF as well as plasma. And there's a lot of information coming out there now in the literature that can at least start to make associations with disease progression and certain types of profiling. So that's what we're planning to do.
Mark, you had something to say?
There's a need for biomarkers for synaptic health, and I see PET imaging agents in development now that might provide such a signal. So for example, to image synapses, one can, there's a novel ligand that binds to SVG2A, a synaptic protein. I think there's a real need for more research in that area.
So we have used blood-derived iPS cells that then we differentiate into neural stem cells as a potential to identify responders, a regenerative responder versus a non-regenerative responder. And so far that has worked out very, very well, except for the time frame. that it takes to not so much reprogram PBMCs into iPSCs, but to differentiate iPSCs into neural stem cells. And that time frame is still a bit too long for incorporation into the clinical trial design to identify potential responders versus non-responders. But that technology is rapidly evolving, where we could in fact use and not be hindered by the temporal aspects. Rochelle, do you have?
I don't have something on the specific biomarker question, but another thought that I would like to raise, and really I think it's most acute, Mark, for you and the work that you're doing. You know, there's a regional nature to Alzheimer's disease, where the disease starts, how it spreads. There may even be inter-individual variation in that specificity. And so, you know, if you're delivering a gene therapy, in particular, you have to start with where to deliver it. You've chosen, you know, the areas of the brain that are affected quite early. And you're choosing a trophic-based approach. What are you thinking? And it also, even, Ali, with your choice of temporal lobe, you know, for your modeling, how comfortable are you that you want to choose the first affected area rather than the most recently affected area with this type of approach?
Yeah, that's a great question. So we are choosing to target the entorhinal cortex and hippocampus early because it is the site of the earliest pathology. And ideally, one would intervene as soon as possible. So ultimately, that could mean intervening at a pre-symptomatic stage. And that's why this biomarker discussion is so important. And we intend to make use of the ATN guidelines that have recently been put forward to use the full spectrum of pre-symptomatic markers that are available as we take the long view of this program. Does it make sense for us to target circuitry that has already been affected? And at the stage at which we would intervene, which is MCI or early AD, 50% of the neurons are still present. So that's the population that one would try to target for protection and functional enhancement. So, you know, I assume everybody here practically sees Alzheimer's disease patients. If we could maintain function or improve function at the earliest stages of clinical Alzheimer's disease and prevent deterioration, I think that could have a stunning impact on the disease, assuming our hypothesis is correct.
Do you think the impact would be higher if you could target the area of most recently affected progression?
Yeah, you know, that, again, the fantastic hypothesis. In the gene therapy world, there is an effort, and we are making an effort to develop new tools for more broad dissemination of the vector. And there are methods, hypothetically, under development where we could target broader cortical regions and indeed address what you're suggesting.
Michaela or Ali, you know, you've kind of targeted in on a region as well. What are your thoughts?
You know, we have some publications out optimizing structural measures in the entorhinal cortex and related areas which are early affected in Alzheimer's disease. And you can see the progression from basically what is BROC1 to 2, that is the transition affecting the entorhinal cortex from lateral to medial with the structural imaging tools that we have. And then we're mapping those to tau imaging, working with Servo, using their agent. And this is a very, very sensitive spot in the MCI phase of disease, which I guess you were alluding to, where there is some very active damage across that BROC1 to 2 phase, and then you can look at the cortex. And so the tools are really coming on board, and we've published a lot. This is all open resource being done by the Center for Imaging Science at Hopkins that is being used in our trial. And you can really, I have a movie on my computer that shows this pretty breathtaking. If you use the 3T imaging data that exists in ADNI, according to the enrollment criteria in our trial, this degeneration sweeps across from lateral to medial entorhinal cortex, and you can very sensitively measure it and pick it up. We hope to slow that down.
Anything?
I have a question that oops, somebody else first. Go
Oh,
ahead.
I was just going to say the reason we decided to start with the temporal lobe in terms of our studies is because we didn't. So other studies that have looked at prefrontal cortex, often a patient will be deceased before the disease pathology moves to the prefrontal cortex, and so we're looking for studies that we are sure that there's pathology in the tissue. I think that's important in doing these large-scale studies is to start at the temporal cortex or the entorhinal cortex so that you know that you're standardizing your tissue and your pathology.
Are you worried that might or might not be the most responsive area once you get it?
Yeah, so I guess it's what Dr. Gallagher was saying, where it just, I guess, depends when you're catching the patient. So if you're catching them at MCI, it might be the most responsive, but if they're coming in way down the road at AD, you're right, that area might be already affected.
I was also going to follow up with Dr. Taubes on just a question about the approach. So I think that your screening approach is based on comparing the differential expression of genes in ApoE4 patients who have clinical disease and not. I'm wondering, did you think, so the risk there is that the asymptomatic patients are just on the threshold of developing symptoms. And you might be trying to discern a signal that could be very subtle or clinically not important if they're about to develop disease. Seems like a comparison to the E3 patients could be very informative too. I mean, there are lots of screens you could do that are the basis of your comparison. So what about that your E4 patients might not be, might be about to develop disease?
Yeah, that's actually something that we've been combating a lot. So we found that what you're saying is actually true for 3-4 patients. E3-4 patients, the control group is not actually a control group. We see them be very close to the AD group, and it's mostly because you catch them before they're 1 or 2 years away from developing AD. But the E4-4 group, because most of them are above 70, they would have, in the average age of onset is 68, you can be more sure that they're not going to get it, that they're somehow resistant. But that we do see that distinct problem in 3-4. I don't know if you noticed, but on my slide, there's not a lot of genes that come up as differentially expressed in 3-4, and we think it's because most of the controls are probably going to develop Alzheimer's at some point.
Another really interesting data set to exploit with that approach would be the Icelandic patients who have the protective mutation and drugs that replicate that transcriptomic signature. Have you thought about that?
Yes, we have. And also we have a large population of E2 that we haven't even delved into. So after meta-analysis, we probably have 40 E2 patients, which is quite a lot given how rare E2 is. And so that's a whole nother area that we're definitely looking into.
So I had a question for Frank to kind of dovetail off of what you brought up, is this dichotomy between systems biology regulators and a strong development in big pharma and in pharmacology writ large around very precise therapeutic targets, one target, one outcome, and your thoughts on the systems, activating systems biology therapeutic targets.
Sure. Yeah, that's a good question. I think, in these neurodegenerative diseases, including Alzheimer's, I think there's so many mechanisms involved, depending at what stage, but even early stages from what we know, that I think more and more we have a picture of really degenerative signaling networks. I mean, I think for many of the speakers today, it's about networks, and I think the targeting single areas has proven to be difficult. Now, maybe if it's the right area at the right time, it could be effective. And so I think we have so many causative potential factors now. I mean, just think about aging. It's the biggest risk factor. It might be even more powerful than ApoE, right? And what does aging mean? There's a lot of mechanisms involved in there, from inflammation, et cetera. So it just, from a biological point of view, just seems so risky to target one area. I mean, we could get lucky. So so we asked, well, we're not smart enough to figure out what to target. You know, how does nature affect broad networks? What did it pick, right? And so I kind of always joke, it designed the receptor as a powerful way to affect, you know, broad networks. So yeah, I think it's about networks on both the intracellular level and networks on the brain level, as some of us are targeting. And then that brings up the really complex area of biomarkers. And you know, what biomarkers does one look at to detect these network effects?
So I'd like to make let's look at what is FDA approved. So FDA, if you actually go into clinicaltrials.com and you're doing informatics analysis, that database is really frustrating to someone interested in informatics because there really is no entry in terms of target. In fact, the FDA generally doesn't approve based on target. It's approved on safety and efficacy. And whereas and when you go for molecular targets, most of them actually have other effects. In fact, I think that was the last speaker was bringing this point up intuitively with basically the systems analysis. So you can have, so pleiotropy is when you have a drug that has multiple things. The good news, bad news about steroids is it's massive, and that's part of the bad therapeutic index. So dosing is dependent not on how much you give, but when you give it in the therapeutic time window and how often you repeat it. So a single target drug can have very different effects depending on whether it's disease modifying or single. In teaching, I always use the fact that aspirin works. So you take 2 aspirin, you get a palliative effect. If you're taking low-dose single aspirin, you're getting a biosynthetic process effect, okay, and it's protective. In fact, the patient can even go off, and yet you're still going to have time. They can go off it for a day or so, and you're still going to be able to recover. So it's the process you're looking at, the dosing, which is more than just the amount, and generally, the vast majority of drugs in current clinical use are not single targets. They're systems approaches. Research scientists abuse them, using them as research tools, thinking they're looking at a specific target.
So to wrap out this session, what I'd like to do is switch it to gears. Since this is the emerging therapeutics session, we're still pretty close to where we began. Well, at least we have the memory of where we began. And so what I'd like to do is ask each of us to give a lesson learned from this whole process of developing therapeutics from inception to, for most of us, in clinical trial. And I'll start out. So the lesson learned for me, one of the lessons, one of many, is to build team skill sets, that from start to finish it will always be teamwork, and to build a team set. Mark?
My message is to learn from failure. So inevitably, many of our first efforts fail. If we design the experiments correctly and we're not zealots about our hypothesis, then we have a great deal to learn from what we've done already that may not have shown efficacy.
I guess I would say I started for 20 years studying aging and neurocognitive impairment, and there was a big wall between aging and Alzheimer's disease. And I think it's welcome now that there is a little bit more attention paid to the fact that late onset Alzheimer's disease is the most common, and that you need to kind of incorporate thinking about the aging brain. It's not a mess. It's actually in pretty good shape, the aging brain. No neuron loss. There's a bright line between aging and neurodegeneration, and that is sort of the translation there is, I think, really important to late onset Alzheimer's disease.
I'm going to do a little follow-up to what Mark said. We need to learn from our failures. We need to start early. I'd also like to emphasize that our approach is going to be to work with the simple systems that we think we understand a lot about, such as early onset, FAD, try to get a comprehensive read on what's going on beyond simply the A-beta 42-40 ratios. We want to really understand how these mutations are broadly affecting the system, perhaps even identifying additional therapeutic targets, taking advantage of AMP-AD and all of the other publicly available data sets. So again, I think there was a really big step that actually Mark alluded to in terms of trying to identify a continuum of Alzheimer's disease based on pathology, not based on simply clinical manifestations. So I think this is a big step for us, those of us especially focusing on the pathology of Alzheimer's disease. And I can't emphasize enough the importance of these early onset FAD models, such as Diane, that are going to help us at least to organize testing in these systems that are perhaps simpler, that we think we really understand a lot more about than we do sporadic AD.
Thank you. Linda, lessons learned.
So lessons learned, I agree with Robbie. The team is of utmost importance, and you have to be patient. This takes a very, very long time. You have to be motivated and dedicated, and you have to be able to write a good grant. Get money to do it.
I think lessons learned at the very, very beginning, first stage of thinking about a therapeutic approach mechanistically, start thinking about the demonstration of mechanism engagement and endpoints and biomarkers of how you're going to show it's doing anything.
Mark?
Yeah, the lesson learned, I think, is that the experiment needs to be done in a patient, that the mouse models are not showing as much translatability as we'd hoped. To be able to do experiments in patients, that means running a clinical trial, and the costs for clinical trials are just too high. What's driving the cost is the low recruitment rate for patients. That means more sites and longer duration. So I think the focus needs to be on how do we find patients and encourage them to participate in clinical trials, and then run smaller trials with fewer clinical sites that complete more rapidly. And that would drive costs down for everyone in this field.
Rhonda?
So I'm new. I don't have much lesson, but I'm still very passionate. So I think while we are very focused on precision medicine genes and the pathways for AD, and at the same time we can step back and look at the high level, like a clinical phenotypic data. For example, how AD is associated with other diseases. Maybe we can gain some unique insight from other diseases, and yeah.
So I'm going to go beyond what Mark and Steve said and say that negative studies are almost never failures, okay? And when I think back to the first lecture I gave on Alzheimer's disease, what we know now versus what we knew then, we've learned a tremendous amount. So we've got to stop talking about failures and start talking about learnings, but we should not go beyond the data, because there are also instances where we have done that, in particular using the biomarkers of AD neuropathology and generalizing them well beyond what they really represent. So A-beta and tau, they don't represent surrogates, they don't predict things, they won't necessarily reflect a treatment response, et cetera, et cetera. So we should not go beyond the data with our enthusiasm either.
Thank you. Alice?
Yeah, I'm going to talk from a training perspective because that's what I know. But I think we really need to be investing in computer science training. I know myself, I had to teach myself, and I wish I had a CS undergrad. And so just getting either CS people interested or giving biologists the computer science training they need, because we're entering an age of massive data. So I think it's really important.
Thank you. Marty?
Yeah, I think the lesson learned is really failing fast and early, and having your animal model, your early studies, don't fall in love with a model. Is sounds like some advice you would give your son, but the thing about it is that pick the choice of the model that has your pharmacodynamic endpoint in it that you can kill your project early. And then at the very beginning, when you're starting out the campaign, find a physician who's a good diagnostician and has an open mind. It is not overly focused, as you mentioned, on pathology endpoints, but actually sees patients going through the progression, because then you get at the clinically relevant pathophysiology progression. That's the biggest thing I've learned.
Thank you. Well, I'd like to complete this session 4 with thanks to all of the contributors. Special thanks to the team that's been working on allopregnanolone with me, and very special thanks again to the organizers, Dr. Susanna Pedincheska and Laurie Ryan. Thank you.